Hi there, so this video is the second part of 20.2 Biotechnology. We're just about here on Objective E and we're going to explain and investigate the use of lactase, which is an enzyme, ASE, to produce lactose, the sugar-free milk. So milk has lactose sugar in it, and if you want to remove the lactose, use lactase to break it down. So, lactose intolerance then. This is when you can't tolerate the sugar lactose. Now, lactose is a disaccharide, two sugars, and it's found in dairy like milk. So if you drink milk, your body wouldn't have enough lactase enzyme to break down the lactose in the milk. And the problem with that is lactose will then pass through your body, the sugar, and bacteria will feed on it. And they create a lot of gas, which causes flatulence, a lot of them, a lot of stomach pain as your intestine expands, and diarrhea can cause that as well, so not a great thing. So lactose-free milk, how do we make it? We take the lactase enzyme and we add it to the milk that's got the lactose in it. The lactase enzyme, you can make it using yeast and fungi with the process of fermentation. Lactose is basically broken down into two things. Because lactose is a disaccharide, it's broken down into two monosaccharides, and they are glucose and galactose. Okay, so with yogurts, when you make yogurts from other dairy products like milk, you can add the lactase enzyme, and what it does is any lactose in the yogurt that's in the dairy would be broken down. Lactose is a disaccharide, and it would be broken down into glucose and galactose, which would make the yogurt very, very much sweeter. If you've ever had a yogurt, sometimes it can taste very sour. Immobilizing lactase, so this is where you immobilize the enzyme, and again it's putting the enzyme on those beads. You then pour the milk through it, and the lactase enzyme would react with the lactose in the milk and break it down into glucose and galactose. Then the glucose and galactose in the milk would fall into a beaker or whatever, and it's a great thing because the lactase enzyme doesn't need to be replaced. It doesn't end up in the milk at the bottom of the columns. It can just be reused over and over again. So the investigation here, basically very simple. All you do is get some warm fresh milk and you test it for glucose. It shouldn't have any glucose in it because it will have lactose in it. But if you then test milk with some lactase enzyme, it should break the lactose in the milk down into glucose and galactose. Therefore, if you tested it after adding lactase, then you should get a positive test with the glucose test. Remember the glucose test, Benedict's solution. You add the Benedict solution and then heat it to a very hot temperature, 80 plus degrees Celsius, and it will turn from a light blue to a spectrum of colours from blue to green to yellow to orange to red, and orange red indicating a lot of sugar is present, a lot of glucose is present. Okay, lastly in this topic we have to talk about how fungus like penicillium, the fungus penicillium, can be used to make antibiotics like penicillin. Antibiotics then, so antibiotics are those things you take when you're sick, when you have a bacterial or fungal infection. They don't work on viruses, but they can affect bacteria because they disrupt the cell wall of a fungus or a bacteria and can destroy them. That's why we use them, because we don't have cell walls. So antibiotics don't really affect us at all, unless you're allergic to them. Quite a lot of people are actually allergic to penicillin, for example. I am, so I don't take penicillin when I need antibiotics. So basically, when we talk about fermentation, we talk about bacteria and fungi making stuff for us, like alcohol, wine, biofuel. But also, they can make other things, like antibiotics. So antibiotics are complex organic compounds, and they're made by bacteria and fungi just naturally. You can imagine that a bacteria might be in a certain area where there are other strains of bacteria that it's in competition with. A bacteria would create its own antibiotics to suppress the other bacteria types so that they can't survive, and so only this one type can. So, yeah, for example, streptomycin, the antibiotic, is produced by this guy here, and yeah, this is just a type of bacteria, and he produces that naturally. So all we do is basically we take it for ourselves. So if you had different mutant species of that penicillium fungus, he could create, essentially, he or she, it could create a different strain of antibiotic, a different type. So what we can do is we can tailor the forms of penicillin that are made in labs to make them more effective. Examples are you can make methicillin, which is a different antibiotic, ampicillin or oxacillin, and these all are made from or derived from the original penicillium fungus that would make penicillin, but then they've been mutated or tailored slightly in the lab to create different forms, which can be used for different things and different strengths. So the effects of them. So there are different types of antibiotics. This one, bacteriostatic antibiotics, they're the ones that disrupt the cell wall production. So the bacteria can't reproduce and it stops them from undergoing protein synthesis. So basically, they can't reproduce, they can't grow. Bacteriostatic antibiotics stop growth and reproduction. Bactericidal antibiotics cause the bursting and death of bacteria. So there's two different types there. Again, animal cells don't have cell walls. So we don't have the same structures that bacteria have to make protein synthesis happen. So the antibiotics don't have an effect on animal cells. Okay, so part G is to explain how the fermenters are used in the production of penicillin. So you get your big fermenting tank and you fill it with your food stock solution with your minerals and sugar. The sugar could be lactose, or you can use this corn steep liqueur, which has basically got amino acids in as well. So this food is going to be used by the microorganism that's creating the penicillin to respire and grow and therefore make more antibiotic, more penicillin. This corn steep liquor, in fact, is a byproduct of the production of corn flour starch. So that's where we get it from. The pH you need is 5 and 6, so it's slightly acidic, and the temperature is standard 27 degrees. Air is blown through, again, adding oxygen for fungi respiration, and it's stirred, much like the diagram we looked at before. So the microorganisms, the fungi, are left to grow for 2 days, which is why it's really important to start the whole process in sterile conditions, because if you're going to let them grow for 2 days, dangerous bacteria, if they were in there, they would grow for 2 days and could contaminate all of the product that you're going to make. So being sterile, being clean at the start is very important. So the fungi are going to feed on all the nutrients being the corn flour, the corn steep liquor, or the lactose, and they're going to feed on that, and the amount of nutrients are going to decrease. As they decrease, at this point the fungi are going to secrete their own enzymes into the nutrient fluid to break down the remaining nutrients to absorb them and feed off them. You then filter off the nutrient fluid and you extract the antibiotics that have been made by the fungi. Okay, and that's topic 20.2 done. We'll now take a look at the last bit, 20.3. Okay, so the third and final part of 20 biotechnology and genetic engineering is to talk in detail about genetic engineering, 20.3. These are the objectives here. And we'll start by defining genetic engineering. Basically changing the genetic material of an organism, like a bacteria, by adding or altering individual genes in that organism. These are some examples: insulin, making insulin, or GM crops. And insulin, basically, people with diabetes don't make enough insulin, or their insulin is faulty. So they need a good supply of it. And normally you'd use like pigs or cows and use their insulin, but it's not always ethically acceptable to use pig insulin, and also it can sometimes cause allergic reactions. So we use genetically modified bacteria. So we modify genetically the bacteria so that they create insulin. How we do that is we cut open the plasmids, insert the genes that create the insulin, and then the bacteria then creates the insulin. GM crops, now the soybean and maize plants are just regular normal plants, but what they do is at the minute in the USA they're starting to add genes to them so that they're resistant to herbicides and insect pests. It's still experimental, it's not been done properly yet. Pest resistance, basically bacteria can produce toxins that kill caterpillars and different insects. So what we do is we add the gene for this toxin to the plant using a bacterial vector. So we would transport the gene in the bacteria into the plant. The plant would then assume that gene as part of its own genetic makeup. So the plants would then start to create their own toxin and have pest resistance, which is great. They also, because it's part of their own genetic makeup now, they then pass that trait down to their offspring. The only problem is insects are going to start developing resistance to that by natural selection, and then all the plants will again be susceptible to the insects once more. Okay, so next we talk about herbicide resistance. Now, glyphosate, I think that's how you say it, will kill any green plant. So you could use it on like weeds because it kills the green material, the plant, but then when it reaches the soil, it becomes inactive. But the problem with that is if you're spraying it on your crops to kill the weeds, it's going to kill the crops as well. So what you can do is give your crops a gene that makes an enzyme that breaks down glyphosate so that it's not affected by it, and essentially they're resistant. You wouldn't give the gene to the weeds so they die, they'd perish. Another one is vitamin production. In countries where they don't have enough vitamin A, in like developing countries, you can put the vitamin A producing gene into a plant. The plants produce vitamin A, the people eat the plants, they get vitamin A. Another one is the oil from the rapeseed that we talked about earlier that's used in diesel motors. Genes can be introduced to change the consistency of that to make it more usable in the diesel industry. Also, tomatoes go soft. Well, let's remove the enzyme that makes them go soft. Let's put a gene in there to stop the enzyme from functioning. Next, we have to talk about genetic engineering in a lot of detail in terms of creating human protein. So let's look at the bacteria. It's a single-celled microorganism. It's got these parts that we should know now. It's got the DNA loop, no nucleus, just a loop of double strand of DNA that has genes involved in metabolism, everyday life. But it's also got these little plasmids, and these are the circular pieces of DNA that carry genes that hold resistance to antibiotics. Another thing we need to talk about is restriction enzymes. These are enzymes that are produced by bacteria, and they're really good at cutting DNA molecules at certain points. It actually cuts them here between the A and T bases in the sequence. We'll talk more about these later. Now, the process of genetic engineering involving plasmids, there's a lot of detail about. I'm actually going to use the pictures to describe it. So here's your bacteria and here's your plasmids. Remember that we talked about how the bacteria creates an enzyme called a restriction enzyme. Well, what we do is we extract some restriction enzymes, not from this plasmid here. That's the blue bit is it's the upper part of an arrow that's just added to this diagram and connects to this diagram here. But anyway, we take out some restriction enzymes, and what we do is we use those restriction enzymes, indicated by the E here, to cut out some DNA from, let's say, a human, a donor DNA. So the human DNA, and we're cutting out the gene, the section of DNA that creates insulin. What we then do is use that same, we collect some plasmids up, and we use that same restriction enzyme that we used here to open up, to cut open the plasmid. And remember that restriction enzymes are the things that cut DNA molecules between A bases and T bases. So what we do then is we use a method called gene splicing, where we add someone else's DNA into a cut open part of someone else's DNA. So we take the human gene that creates insulin and we add it to the plasmid here, and then we use a ligase enzyme The ligase enzyme is going to help this to seal together. So we've got restriction enzymes cutting open DNA, and then we've got ligase enzymes helping these to stick together and become one DNA loop. We then put these plasmids back into the bacteria, and they're going to start creating lots of insulin. What we do is use a culture method with antibiotics to select only the bacterias in a big, because this is just one bacteria, but imagine loads and loads and loads of bacterias, and we can select the ones that just have the recombinant DNA in. Recombinant DNA is where you have two sets of DNA in one DNA section here. So a plasmid with this extra gene in from the human is an example of recombinant DNA. So we can select those that have the recombinant DNA, let them reproduce more and more to make more and more insulin, and that's basically the process. So the other methods, basically, you can use a virus rather than a plasmid to be the vector. So you put the virus into the bacteria, or you could just insert the DNA directly without using the plasmid or the virus. Or also, the donor DNA from the human, you could just make it rather than taking it from the cells of a human. You could just make it using nucleotides and fix them together. You could use yeast rather than bacteria. Okay, the last thing to talk about in this whole topic is to talk about the advantages and disadvantages of GM crops. We all know what GM crops are by now, genetically modified food, and we use it. We use the genetically modified food because they have increased yields, so we're going to make a lot more money on our farming products, and we can use it to enhance their preservation. You can notice this in foods on the shelf, you know, those sort of foods that you leave there for ages and ages and they just don't go off. That's because we've genetically modified them to do so. So it's a good thing, and that's the advantages. However, there are hazards of GM food. One of them is antibiotic resistance. Imagine you were using a vector for antibiotic resistance, and whilst delivering the recombinant DNA, so that DNA that has two strands of different DNA in it, or two types of DNA in the strand, if they were to get into harmful bacteria in the small intestine, that bacteria could become resistant to an antibiotic. So there are dangers. Also, pesticide residues, i.e. toxins, could be on the GM food that you're producing. However, you know, there are precautions in place to stop this from harming us. Golden rice. This is a type of rice created in third world countries, and it's rice that has a gene for this chemical here, beta-carotene, which goes on, it metabolizes into vitamin A production in the body. So it's great because it reduces the instances of night blindness, but the problem is it could turn into a toxin once eaten. Another problem is it reduces biodiversity because you're messing with the genes. You're almost selectively breeding them because you're choosing genes that it has and then breeding only the types that you need, like ones that create insulin or ones that create high resistance to pests or whatever. So it definitely reduces biodiversity there. So that's the topic complete.